It's a pleasure today to welcome David Foster from Johns Hopkins University. David is here as part of our series on cracking the neural code, which is sponsored by the Center for Cognitive Neuroscience. Unfortunately, Jim Haxby, our director, can't be here today. But nonetheless, I'll do your presentation. David is from the UK, so he earned his bachelor's degree from Imperial College in London. He received his PhD in 2000 from the University of Edinburgh, where he worked with both Richard Morris of the Morris Water Maze fame and Peter Diane. And he received his degree in computational neuroscience. He then went on and did a postdoc with Matt Wilson at MIT, and then joined the faculty at Hopkins in 2008. Now, the first sentence of David's website, I actually happened to look this up, says, and this is to quote, says that my lab focuses on how large populations or ensembles of neurons encode and process information in awake behaving animals. And I thought this was very apropos for our series of cracking the neural code. David combines work both with physiology and computational approaches. And as you're going to see and hear about today, he records from over 200 neurons at a time, simultaneously in an awake, freely behaving animal, tries to correlate the activity, understanding what that code is actually doing. So the title of his talk today is Neuronal Sequences in the Hippocampus for Memory and Imagination. So please join me in welcoming David.
So,
just turn down this
yeah.
light.
Right. Well, thanks, Jeff. Thanks for inviting me, and I've had a just terrific time today meeting with people and lunch with the students. It's awesome. So yeah, I'm going to talk about the hippocampus, and I get the feeling this audience will be pretty familiar with things like place cells and, let's see, spatial memory tasks and things like that. So I'll try and go through quickly because there's quite a lot to get through. But I want to, so I am going to be talking about hippocampal place cells and telling you things that place cells do that you might not have realized that they can do. But I'm going to start with a rat's eye view of the problem that people think hippocampal place cells are a solution to, which is the problem of navigation. And here's a real maze. You know, you're used to looking at things like water mazes. This is a real maze where you go in and you don't know what you're doing and you've got to somehow figure out how to get to the food. And I want to point out a couple of things that are difficult about this. The first thing that's difficult is the direct bearing to the food from the starting location doesn't tell you very much. Because the actual decisions you need to make are almost unrelated to this direction. In fact, there is a relationship, which is that the vector sum of all of these moves that you have to make does add up to this vector. But that's an extraordinarily weak constraint because there are an infinite number of sequences of vectors that would have the same property. Another example might be if I want to get out of this building, I have absolutely no idea, but it's probably, I mean, it could be here or here. I have no idea where the exit is. But it's irrelevant because I need to take that action. The second problem is to do with associative learning. So if I just want to associate being a being in this location or some actions that I take near to this location, that's not going to solve the whole problem of all the things I have to do from far away. So people have used this kind of a task to suggest that animals and people must have some representation of the world with some degree of complexity that can be used to formulate plans and to figure out how to navigate. So I'm going to explore that idea with respect to neurons in the hippocampus. And this is just showing the rat hippocampus. And I'm particularly interested in these cells, the workhorse experimentally in the hippocampus, the CA1 pyramidal cells. And I'm going to be dropping electrodes into the hippocampus, recording from these cells in freely moving, freely behaving rats and mice. I'm sure everybody's familiar with the technique, but I use a technique called, it's extracellular recording, and I use tetrodes, which are very small microwires of the order of 10 or just over 10 microns in diameter, 4 wires wrapped together so that the tips are very close together, just a few tens of microns apart, and they're recording from the same population of cells. So we can triangulate the signals to separate out the spatially, the different sources, the different cells that are giving rise to action potentials. In this way, I'm going through this very fast, I'm sure everybody understands that you can isolate different physical sources of the signals which corresponds to different cells. And that's the trick, where we can go from a single tetrode to recording multiple units off of that electrode. And then what we do is we implant lots of these tetrodes. And in some of the experiments I'll tell you, we actually have up to 40 of these tetrodes. So we have 160 channels in total to record as many neurons as possible. So it's well understood that in the hippocampus and nearby regions, there are these representations of allocentric space, of external space that an organism walks through or runs through. And so the classic long-standing example is the hippocampal place cell. So these pyramidal cells, each cell will fire in a localized area of the environment. So what you're looking at in black is the search pattern, the behavior of an animal over the course of about 20 minutes in something like a 2 meter by 2 meter arena. And in red, you can see the spikes from one neuron. So they're spatially localized. And we call that the place field. But one synapse upstream, you have neurons in the medial entorhinal cortex, which are called grid cells. And there, a single neuron will fire with this extraordinary lattice-like firing field in space, which is a hexagonal grid. And different grid cells will fire at different spatial phases and at different separations between the grid nodes. But there's this very regular representation of space. And then, I probably don't need to tell people here, but there's the head direction cells in many different areas that have a very strong signal that represents something like an internal compass. It's not the absolute north that they care about, but they have, it's as if in any environment they select a particular direction to be north, and then different neurons will be tuned to different heading directions in that environment. So across these and other areas that have other firing characteristics, there's this idea that there's a real representation of space. So I'm going to talk about what this might mean in terms of navigation. Just thinking about this task again. And what might you want to do with a set of place cells? Well, you know, you're in this environment, you might want to ask yourself, let's say you find some food, you might want to say, well, where have I been? How did I get to this location? Or alternatively, you might be in the middle of the maze and you're trying to get out. And you ask yourself, how do I get out? The key thing about place cells is they tell you where you are. So as a map, they seem to be stuck in the current location. Because by definition, they only fire when you're in the place field. So this is what's been difficult, at least for me, to understand about place cells as a map system. But it's also true for grid cells, and it's also true for any other spatial representation in terms of their field representations, that they fire by definition within their receptive fields. Now, it's been known for a long time that there are two states of activity in the hippocampus. As you can see in the hippocampal local field potential, there's one kind of activity that you see, a theta rhythm, 8 hertz theta rhythm, when animals are running around. As soon as the animal stops running, in this case it's a rat, as soon as the rat stops running, the theta goes away, and you get these periodic, what are known as sharp waves. So see, it's interesting because all of the place field, everything we know about place fields has been gathered from this behavioral state. But this is quite a different behavioral state. So this is known as a sharp wave on this time scale of this is about a minute. And when you blow this up, you can see these high frequency 1 to 200 hertz oscillations riding on the top of it. This is known as a ripple. So this whole thing gets called a sharp wave ripple. So I'm actually going to, everything I tell you about is essentially about what the place cells are doing during sharp wave ripples. So the plan of the talk, I'm going to talk about sharp wave ripples and something called place cell sequences. And they're going to explain that both sharp waves, ripples, and place cell sequences capture something about the shape of experienced environments that I believe must be learned. I'm going to explain that these sequences are actually NMDA dependent, further proof that there's something is learned. I'm going to make the case that these sequences represent a kind of goal-directed memory retrieval that you will see offers some kind of solution to the navigation problem. And then I'm going to end with talking about the possibility of diseases of sharp wave ripples. So we'll just jump right in with the recordings. So this is 20 minutes in the life of a rat running up and down a linear track that's about 1.5 meters long. And you can see in grey he's sweeping from one end to the other, but he's also pausing at each end. On the top in red you can see the times and positions where one hippocampal cell fired spikes. You can see it only fires when the animal rounds the corner. That's a place cell. We can capture this place field in 1 dimensions as basically a histogram. It's normalized by occupancy for those who care, but it's just like a histogram. And if we record several of these cells at the same time, we find that they tend to tile the entire experienced environment. So there's a cell for every location. They're not perfect the way that perhaps grid cells are or head direction cells, but they seem to cover the entire environment. So these are recorded simultaneously. So now let's zoom in on 1 minute during this task, where a rat is running from one end of the track to the other and then pausing. This takes a few seconds, and this is the rest of the minute. If we just show the raster, the spikes from each of these 19 cells, so they're recorded simultaneously, right? And this is time. You can see the animal runs through the place fields. The cells fire in their place fields as you'd expect them to. But when the animal stops at the end, you get these characteristic population events where all the cells seem to fire together. If you zoom in on this, you find that these events are actually sequential. And in this case, it's quite interesting. These events start at the end of the track and proceed in a sequence all the way. So these are the neurons that fired at the end of the track. They fire first, and you get a sequence that proceeds all the way back to the beginning of the track. So the animal's stationary at this point, right? But the place cells are firing in a sequence that corresponds to his previous trajectory, in reverse order, interestingly. And this whole thing takes just 100 or so milliseconds. And this is what happens during the sharp wave ripple. It's a very robust thing that happens while the animals are fully awake, and you get lots of these events during the course of a short 20-30 second stopping period. So this is after the first, second, and third laps on a novel track. And you can see each of these has the same x and y axis. It's about 200 milliseconds, and these are the 19 cells. And the events are very almost stereotypical. So I've taken a side because I know some people are interested in reinforcement learning. Why would you want to have reverse sequences? This is a question I sometimes get asked. Well, one reason you might want to do this is to develop some kind of expectation of the goal that could guide you in navigation. So this is just an we just dreamt this up. This is a scheme for which there's no direct evidence. But imagine if every reverse replay co-occurs with a fast onset, slowly decaying reward signal, then these two signals impinging on some downstream area will allow them to be associated so that place cells, these are the place cells near the goal, these will be associated with high levels of reward, and the places further away will be associated with low levels of reward. So you could actually end up developing some mapping from your place cells to a slowly increasing representation of reward. And this ends up doing something which in reinforcement learning is known as finding a value function. And it's actually mathematically quite well defined. And the point is that it actually solves difficult global navigation problems. So this is an example which you will find in textbooks on dynamic programming and Markov decision problems, which is you want to solve a problem, you want to solve a maze like this. Well, you start at the end and you just count the number of steps. See, 5, 6, 7, 8. You flood the number of steps out from the end, but you must do it in reverse order. And if you do this, you end up with a set of values for each place that renders the navigational problem completely local. Because at any point in the maze, you just have to take the step that will take you one step closer to the goal. We can talk more about that later because because I have a lot of data to get through. But that's actually to me it's a very interesting idea that you could actually have reverse replay could serve this interesting learning role. But both reverse replay and forwards replay in the awake state has been replicated many times now and extended. So as I just said, you get forwards and reverse replay. You can have replay sequences that extend over very long spatial tracks. So here's a track that's about 10 meters long, and the replay will just happily chug along at a speeded up rate, but at a constant speed. So if there's a replay that depicts a 10 meter long trajectory, it'll just take 500, 600, 700 milliseconds to complete. It's kind of amazing. So this shows this nice relationship between the duration of these events and the space that's being depicted. And it's really clearly correlated. And there's some work suggesting that these events are necessary at decision points for animals to be able to choose, for example, in this alternation task, at this choice point, they have to have these events, and if they can't have these events, then they can't do the task. Well, I'm going to expand upon that later. So these sequences aren't going to be any use unless they capture the navigational structure of the environment. I would posit that. But how can we test this? Well, one thing we did was to take an unusual shaped environment, this Y maze where one arm is twice as long as the others, and show that we can just examine what kind of replay sequences we get on this track. So here's a linearization of position. So I've taken these arms and projected them onto a line, and you can see that there's this behavior which corresponds to the animal, let's see, starting here, moving down along the Y arrow, moving back to the center, moving into the green arm, moving back to the center, and moving into the purple arm. Okay. We recorded over 80 cells with fields along this track. And as you can see, we've aligned the cells according to where their peak is along the track. But it's quite messy because some hippocampal cells fire in more than one place. So just picking one position isn't necessarily that informative. And also, the distribution of the place field is informative. And so just picking the peak, it doesn't tell you very much. So we actually make extensive use of what we call decoding, a decoding strategy, which is basically, it's a very simple statistical strategy where we take each of the cells' spike rates and each of the cells' place fields, and we say, what's the most likely position for the animal to be in at any given moment, given these spike trains of place fields? Actually, we don't even do that. We calculate for every single position a posterior probability that that is the position that the hippocampus is depicting. And as you can see, that nicely captures a very tight prediction at every point. So the very first time an animal goes onto this funny shaped Y maze, in very short order, the system starts producing replay sequences. So these are just some of the examples, it's not all, of the replay sequences that you'll get within about 20 minutes. Well, this is a bit long. It's within about an hour of experience on this novel track. and you get replay sequences that correspond to each of the arms, each of the two arm trajectories that involve going from one end of an arm to another. Does everybody know what that means? So you've got three ends. There are three possible ways to get from different combinations of ends to other ends, and every single one of them is replayed in a very strong way at some point. And this actually is a bit hard to see, but at different stopping points, you actually get many replays, and replays of different trajectories. And in fact, we see replay very quickly. So this, what you're seeing here is that counting the number of significant replay events and comparing it to the number you'd expect by chance if just the cells were firing randomly. And what we find, just focus on this red to blue to red depiction of significance. Basically, we find significant amounts of replay very quickly, and after only one or two experiences of the individual trajectory. So an animal has only to run up and down the trajectory once or twice, and you can immediately start replaying it. So your first thought might be, well, that's interesting, but how do we know there's any learning? I mean, maybe there's just some, maybe these trajectories are encoded as elapsed time, or some representation of experience that doesn't require any spatial knowledge, and we've just mapped it onto space with this decoding strategy. How do we know that this thing is really spatial? So this analysis turned out to be quite important. We say, okay, imagine the animal sitting here at this, what we call the C end, and he's spitting out replays. Some of them go up here, and some of them go up there. Well, are they using the same cells to code for the common portion of the environment, as in this case? Or are they using different populations of cells to encode for the different experiences? This would be easy to generate quickly, essentially without learning, because you can take pre-existing sequences and just map them onto the new environments, but just you essentially don't need to learn anything in this case. This would involve stitching together the sequences in a manner that really captures the topology of the environment. So what we did was well, let me back up. We look at the firing rates. There are, let's say, these 82 cells firing during replay sequences. And you can see that some cells fire a lot, and some cells fire a little. And there's a lot of variation to the extent to which these different cells take part in these different sequences. However, there's no variation at all in whether it's a sequence that goes to the left or to the right. In other words, you cannot tell that the same population of cells takes part in this part of the replay and with the exact same firing rates, regardless of whether the replay is going to then move up here or move here. So, and we verify that by just shuffling the identity in the end between left and right going sequences. This is kind of a complex point, but we believe that the evidence favors this mapping in the brain for generating these sequences over this. So we think that this is the kind of thing that couldn't be guessed before experience and must involve some kind of learning. I'll give you one more piece of evidence that favors that. So we also find that in these joint replays that span the arms, the local field potential, the ripples in the local field potential have an interesting structure as well. So this is the raw trace, but what we're looking at here is the power, the ripple power, in this event here. So here's a replay that moves smoothly down one arm and into a second arm. But you can see that there's a peak in the ripple power that falls to a minimum, and then another peak on the other arm. On the other hand, we sometimes see these single arm replays. And you see in these examples, there are no such peaks. So across all the data, we find, particularly when there's a long arm, that there is a dip in the ripple power at the point where the replay trajectory crosses the choice point. In other words, we can infer from the ripple structure alone something about the structure of the environment that the animal's experienced. In this case, that it has a single choice point. So that's the most complicated part of it. That's always a little complicated to explain. But I do think that it suggests that the topology has been captured. Now we're taking a very different strategy. If sequences are learned, we should be able to block synaptic mechanisms of learning and get rid of them, right? This is actually a really interesting question because place cells react to blockade of NMDA receptors and therefore NMDA receptor synaptic plasticity in a very strange way. Actually, they don't. So first order of approximation, they don't react. So this is a classic result from Cliff Kentros and Eric Kandel some time ago, almost 20 years ago, where they showed that you can give a drug which blocks NMDA receptors systemically and blocks synaptic plasticity in the brain and place an animal in a novel environment, and place fields will form and be more or less stable for up to an hour. And only when they record 24 hours later do they find that in these animals, unlike control animals, that there's a remapping. So they don't hold on, the place fields no longer fire in the same place. In a normal animal, the place fields will fire in the same place. I will skip the next point. Why is this important? It's important because we know that spatial learning doesn't work like that. And this is an experiment that was recently published by Richard Morris's group, which nicely shows the performance of an NMDA receptor blockade group and a control group in just a bog standard water maze task, reference memory, learn the single platform in a novel environment. But in this time with enough trials that you can actually see performance reach an asymptote. 4 trials in, and control animals are performing as well as any animals can in this novel environment. And indeed, each trial is a minute or so of experience. So they've had, each trial is this much experience. So they've had about 2 minutes of experience spread out over about 10 minutes. Over that time scale, animals with NMDA receptor blockade are completely unable to learn. Place cells with place fields have formed, we can infer, and are perfectly normal throughout this. So what's going on? Maybe something else. Maybe we can find something else that breaks under NMDA receptor blockade. And you can see where I'm going. I'm going to say I think the sequences are the thing that fails to form, even though the place fields are more or less fine. So to test this, we have this experiment, this timeline, where we have the animal being exposed to simple linear tracks, a non-hippocampally dependent behavior that our NMDA receptor blockade drug won't affect. We expose them to novel tracks, both before and after giving a systemic NMDA receptor antagonist or a saline control. And we look and see what happens. Well, we partially replicate the Kentros results. What I didn't tell you was there was sort of competing literature where people have found small effects. So Cliff said there was no effect for the first hour. There were some NR1 knockout mouse studies that suggested that you could have effects of knocking out the function of NMDA receptors, although that was a knockout mouse, so there were the usual attendant qualifications for whether or not it was a developmental effect and so on. We come up with a result which is somewhere between the two. So here's, in blue you see typical place fields. These are all just different cells. Typical place fields for under saline and under CPP. And this is before drug and this is after drug. And the drug slowly kicks in and is actually at its strongest effect in run 3. And what you can see here is that the size of place fields does get a little bigger under drug, And the sparsity, this measure, actually it should be called diffuseness, but for historical reasons we say sparsity, the measure of how diffuse the place field is on the track does increase a little bit also. But the critical thing is our ability to decode space from this population of cells is barely affected. And you can get well above chance decoding with these slightly bigger fields under drug. So in the spirit of what Cliff Kentros found, CPP is not is slightly smushing some of the place fields, but it's not removing a substantial amount of spatial information. And in fact, position can be decoded from these place fields. But replay is a different story. So the top rows are replays, actually the best correlated replays from saline animals, and in the red, CPP. And what you can see is the drug, these are the best. So the drug completely obliterates replay, these awake replays in the CPP case. And this is an interesting dissociation because we've got the animal running up and down the track through place fields which are more or less fine and acting in a fairly coordinated way, at least, to be able to decode position on a behavioral timescale. But every time the animal stops, it's just garbage. And this is a shuffle analysis which proves this effect, but you can just see it. So there are still two possibilities. Maybe an interesting one and a slightly less interesting one. The slightly less interesting one is that maybe NMDA receptors are just interfering with the ability of the system to generate this more precise, these fine-timed 100 millisecond long events. The interesting possibility is that the animals learn something during experience, and that learning is necessary to produce replays. So we look in a subsequent sleep session, and we find in saline, you can get replay of, in fact, all three preceding experiences at different moments over the course of several hours. We'll find different replays corresponding to different experiences. And you can see here, this is our shuffle analysis. You get significant numbers of replays of the first experience, for example, and significant numbers of experiences of the last experience. The last experience actually looks a little bit better. But in the CPP case, as you'd expect, the replay of the most recent experience that was experienced under drug is completely gone. But there's plentiful and excellent looking replay of the experience that occurred before the drug was given, even now under drug. So the drug isn't interfering with the ability to produce replay, it's interfering with the ability to learn something from experience, which allows you later to produce replay.
Okay, so runs 1, 2, and 3 are in different tracks.
Yes, and can be distinguished in the later sleep session.
And how many cells do you have to be recording from in order to be able to pick out the cells that have these patterns?
Well, so we're looking at all the cells, and the patterns are across all the cells because all the cells are feeding into our decoding procedure, but we're averaging something like 80 cells with fields.
You have a sense of it. There aren't cells that you record
No.
from that are not shown.
No. Just to illustrate the effect as a rate, this is in the sleep. So I say most recent experience replays best. So blue is saline, right? So the most recent experience is replaying at a fairly high rate. This is noisy because it's still hard to pick out replays. Remember, even with 80 cells, the more cells you record, the more replays you can detect. And we've chopped this up into small time bins so you can see a rate. But you see it's a fairly constant rate across, about 40 minutes of replay in the saline case for the most recent experience. Less so for the previous experiences, but still significant numbers. But in the CPP, you get no replay of the two experiences experienced under drug, but essentially maximal levels of replay of the experience that was experienced before the drug, and interestingly better than in the saline case. So the cute way to put this is, for the CPP guys, this was the last experience.
And this is done during sleep?
Yeah.
And what stage of sleep? I mean, that's
So this is actually during we call it sleep, it's during a rest, so the animal's in a different environment. It's been taken out of any of the tracks, is in an enclosed sort of environment, but it's really just a rest box. So they curl up, but we don't know how, we're not claiming that they're in at a particular depth of sleep. Just as a control, we can take the worst fields in the saline case that all have a diffuseness that is at least as bad as the, I think, median level from the worst CPP group, and it doesn't make any difference. So it's not the place fields per se that are responsible for this. There really is a failure of the coordination in replay. So what I'm saying is, I think you get place fields for free, but when the animal runs, he learns something about the connectivity between the places that enables you later to retrieve that in the form of replay. So let me push on. Retrieve that as in the form of replay. So what am I saying? I'm saying this. We think place cell sequences may be a kind of memory retrieval. So that's what I'm going to talk about next. I think it's fair to say that it's been difficult in the field, in the place field, to get a good handle on whether place cell sequences can reflect goals or remembered information about the environment. I mean, some people, including those present, have done pioneering work on this, but it's a difficult question, and there are two reasons. One is that, consider a water maze. The statistical power of a water maze is that there's one small little platform hidden in a huge, great big pool of water. And if the animal can go there, then you have a pretty strong belief that he's remembered it. But if you have a T-maze or something, then, you know, the number of trials you need to get significance with this binary choice is huge, and then there are other factors like fatigue and so on. That's why T-mazes were so hard for so long to get a good handle on, to really prove that there was spatial memory, and then the water maze, I think, was very useful in that regard. That and a number of other reasons like Q control, but anyway. So we needed a spatial task where the goal could be any one of 36 little wells of chocolate in a very large, well, relatively large 2 meter by 2 meter arena. But there are two problems. Firstly, that means we needed place fields everywhere. Now, if you think that to do a replay experiment on a linear track, you're going to need between 10 and 20 place fields in a row. That means, roughly speaking, in an open arena, I'm going to need between 10 squared and 20 squared place fields, right? We sort of ended up landing in the middle of that range, but that's a lot. The second problem is just experimental design. If you get a task where the animal learns to do something, like get to a food location, let's say this was designed exactly like a water maze. Well, then I'd have lots of trials like this, where the animal gets released at the side and just runs straight to the food. And you're not getting sampling of the entire environment. You're getting this very reduced sampling of the environment because the animal is doing something in a very goal-directed manner. So we developed a task that would toggle between two different kinds of exploration. Goal-directed memory-based navigation and random foraging. So here's the task. The animal has to, let's see, on odd trials, he doesn't know where the food's going to be. He's got to search for it. And he finds it. The next trial, he can go straight back to the remembered location. Then he's got to wander around again. He finds some food. The next trial, he can go straight back to the remembered location. And he toggles between these two things, random foraging and goal-directed navigation. We know that they remember where the food is because it's much faster getting to, and takes a much shorter path, both getting to for the remembered trial than the random foraging trial. And note that one nice aspect of this is that the shortest path is the same in each case, right? So if he goes from here, out, or back, the shortest path between these 2 points is the same, right? So we know that the animal can't smell the food, because if he could smell the food, he'd be just as good on the random trials as on the home trials. We know that he cares enough to do it because he's faster, etc. So the second problem we had was the number of cells. So we miniaturized everything. I had previously put in 20 tetrodes to the right hippocampus. Now I decided to put in 20 in the right and 20 in the left. We 3D print our drives, and we were able to get the weight down. We just shaved off everything else, make it as small as possible. These are actually not these are little chips, so they're actually not very heavy. This is not very different to the size of what I was implanting in the rats as a postdoc, but that was 18 tetrodes. This is now 40. And this is what we get for our trouble. So this is not the best session. The best session was actually 250 cells with place fields. And actually, we've just been looking at the data some more, and there's another session that's 263. So I think that's the limit, 263. But this is an example of a day where there are 212 cells. Each of these is the place field, right? By definition, it's the firing rate of the one cell as a function of location in the 2x2 arena, right? So here's a cell that fires kind of on to the left. Here's a cell that fires to the right, and so on, right? These guys are inhibitory interneurons. They fire everywhere, but it's easy to identify them just on the basis of the waveform, spike waveform width. Let's zoom in. These are sort of typical fields. See, some of them have firing in other places, but many of them fire in one place. The size of the fields is interesting. They're pretty big, right? I mean, a rat might be 10 to 20 centimeters, and these are more like 50 centimeters in width. What are we going to do with all these cells? In a way, I wish I'd said this at the beginning, because this explains the decoding strategy a little bit better. For aficionados, we're just calculating a posterior probability under two simple assumptions that we know are wrong, and we want to see deviations from this. So that the cells are all independent, and that the only thing that governs the spiking is the firing rate. If the only thing that governs the spiking is the firing rate, that means it's Poisson. So those are our assumptions, and it's very easy to generate a posterior probability of position from that. But the intuition behind it is that we're looking for the overlap in the fields of cells that spike. So let's see, we have 4 cells that each fire 1 or 2 spikes with these fields. Well, I'm going to have a very strong belief that the animal is here, right? What's cool about this technique is that it doesn't have to be where the animal actually is. If these 4 cells fire 1 or 2 spikes, I'm going to have a very strong belief that the animal's here. The belief's going to be wrong, but it's interesting. You'll see what that means. And I don't always get a position, because if these 4 cells fire, then I'm not going to have any strong beliefs at all. So with all that said, let me show you a movie of the animal running around in this task. And well, I guess before it goes, let me explain what you're looking at. This is the arena. These are the 36 possible food wells where liquid chocolate can bubble up. This is the depiction of the rat's body, his nose, and his tail. This is the home well that's consistently rewarded, though this is actually the random well. Time is going by here, and you see this is in real time. And underneath all of this is this flickering bright light. And actually what that is every pixel on this screen has a probability, and we've depicted that probability as a brightness level. And the fact that it's only bright here is because it's only these positions that are being picked out by the population activity. So let's watch this as it moves around. though the computer has to warm up. So you can see as the animal's running around, the activity of these 212 cells is pretty consistently reflecting his current position. And remember how big those place fields were. So these cells are really combining and coordinating their activity to represent a much more precise location. And then you might have seen, I'll just play it again, you might have seen some interesting stuff happening where the activity seems to move away from the current location. I won't deny that's interesting. That's almost certainly related to the animals in theta at this time. We're not decoding on a fine enough timescale to pull out the meaning of that flickering, but that's other work that we're doing, and you certainly can do that. So now the animal's parked over here, and the question is what happens next? So this is what happens next. All right. So now what you're looking at is a succession of 4 sharp wave ripples that occurred one after the other over the period of just a few seconds while the animal was parked in this corner. The only difference to the previous movie is that this movie is going 20 times slower, and it also relies on a 20th of the data, right? Because our time bins are now 20 times smaller. We're looking at, I think it's 10 millisecond time bins. Before they were nice 250 millisecond time bins. So we've got far fewer spikes. So you expect it to be noisier, but I'll just play it again. But what you can see is consistently in each of these events, the representation of position starts in the current location, moves off smoothly in a sequence, and ends at the remembered goal location.
Why did he pause there? Any particular reason?
Well, he's actually, so that particular one wasn't, right. So normally he's pausing at the random wells, where you can see other movies. There are other places he sometimes just likes to hang out. And this bottom corner was one. But if you saw the amount of time, it was, I can't remember, it's just a few seconds. It's like 10 seconds or less.
Before he moved. So is that at the end of the trial? Or is that
Let me look. So that was, no, it's just right in the middle.
So he hasn't collected the food yet from the green well, the random well?
Well, in this case, he should be going here. So he's not totally on top of the set learning aspect.
There's no reason for him to think that now is the time that I need to go to the green
Right,
well.
in this example, that's right. Although there's a tendency for them to do that when it's relevant, but they also do it other times. So yeah, it was about 4 seconds.
I mean, would you expect that after he got to the random well, because next I want
Yeah.
to remember.
So you get it more at those times, but you also get it. So you also get it at all other times, and you get. And it's interesting. It can also. It's strongly predictive of what he's going to do when he doesn't go to the goal. Also, so it's not a rigid sort of memory signal. It looks more like a planning signal than a memory signal.
So what kind of editing was it? Are you showing us every SWR that was occurring while he's sitting on the
In that case, I was, yeah, within a period of time, yeah. So this is just saying, trying to pick out what's going on here. Individual frames are very tightly focused, so the cells are coordinating to represent locations, even when they're distant locations where the animal is. And then there's a sequence to it, which we haven't imposed. The way we've done the decoding, all these locations could have been in any order. I mean, what I mean is we don't have any history term or any prior moving prior here or smoothing to make this happen. The sequence is a property of the system. You can see there are many different kinds of sequences. This is just to show sort of the examples of the kinds of sequences you can see now collapsed in time, because they're clean enough that we can just collapse them. And this figure, so yeah, he's doing a task where he has to use his memory and he makes his plans. What's interesting about this, so it's an interesting question whether this is really memory or planning or whether those things should be differentiated. But I'll just show you this one way of looking at the data, which is that if you take all of the events that occur when the animal is at the home well, and you just superimpose them, they have in common the home well, because there's a tendency for replays to start in the current location. If you take all of the replays that occur when the animal is not at the home well, which is usually at random wells but also can occur in other places, they also have in common the location of the home well. And if you quantify this, you actually find that across all of the 36 wells, the home well is the statistical outlier. So what this means is that as an experimenter, I can look at the activity of hippocampal cells when the animal's not at, is stationary and not at any particular location. And okay, I have to average across 20 minutes of data, but I can tell you what the remembered goal was in this task. So I think that's interesting because we haven't had that before. It's not because one location was particularly familiar. It's not because the home location was familiar, because there are these locations where the animal does actually like to hang out, and they're actually more familiar. He spends more time there than a home well. But if we look at just group locations according to whether they're more familiar or less familiar than, the animal spends more time or less time than the home well, we find that there's really no difference, and it's only the home well that has more representation. Now, what I didn't tell you at the beginning, I'll tell you now, is that every day we actually move the home well to a different location. The animal has to figure it out for himself, but he has to learn a new fixed location, right? So every trial he has to kind of figure out what he's doing, but the location that gets repeated every second trial is different from day to day, and it gets moved to a novel location. But you can see on the next day, we're also able to pick out the next day's location too. So this is not something about the particular location on this day one. It really is a feature of the goal.
It's a general property that these sequences go from where the rat is standing to some other location.
To some other location, exactly. Yeah. There's a very strong bias. So we know that they start in the current location. We know they kind of end, many of them end at the goal. And then the question is, what route did they take to get there? Because you could still have a very circuitous route. In fact, you could have, if you've taken a long, windy path to get to the random well, you could reverse replay that whole thing and still get those results. So we needed to show that the path taken is actually directly, or at least matches what the animal's about to do, which hopefully, which I already showed you, tends to be a pretty good path from the random well to the home well. So we have this kind of angular analysis. So black is there's an example. Black is a replay trajectory. Green is the future path. Pink is the past path. And we look for the mean angular displacement at these radii along the minimum, the least of the two possible. And what we find is the match to the future path is very good, much better than chance, very strong. You see most of the events are within just a few, have a mean here, angular separation, which is, I can't see what that is, but it's, this is pi. Is that what it says? Yeah, so this is like within 90 degrees, probably less. For the past path, it's not nearly as good. So it tends to match the future path, right? And then this is what Jeff was talking about. So the tendency to get a match is actually better when you're at the random well and about to go home than it is when you're at the home well and about to go somewhere else. My interpretation of this is that it's not that the animal couldn't predict what he's about to do, but that he changes his mind. I mean, he doesn't know where the food is going to be, so he makes a plan. He probably starts off on that plan and then changes his mind because it's not working out. That explanation is as good as any other at this point, but that would be one reason why you'd get poorer prediction at the home well. And then this is quite interesting. It looks like you get an anti-prediction, an anti-correlation of the direction for the past path at the home well. Now, another thing I didn't tell you is the random wells don't repeat within the day. So this may reflect some knowledge that the system has, that the direction of the next random goal is not going to be well predicted by the past one. So the final thing I didn't tell you about this experiment, well, I did just tell you. So every day The goal, the homeworld is in a novel location. And within a day, the random wells don't repeat, at least for the first 19 trials, right? So I'm just talking about that's plenty. For the first 19 trials. And they're unpredictable. So what does that mean? It means that we've kind of got an all-to-all navigation problem, and for at least the 19 trials of every day, every single trial is actually a novel trial in terms of the start and end location. In other words, anytime the animal is at the random well and has to go back to the home well, he's never actually had to start there and go there before, for every trial.
So the trials simply, each trial starts as soon as he's eaten the food in the previous well?
Yeah.
So
Yeah.
nothing else happens? He eats
Nothing
the food
happens,
and then
yeah.
it's-
It's up to him. Yeah. So in fact, all the movies relate to this, but this is an example which you're going to see runtime and replay time activity. So we're going to move from one to the other, all mixed in. But it should give you an idea. This is showing you that he's going to get here, and then he's never had to start from here and go there before. And that was the complete sharp wave. Now this is an interesting one. It goes down here. You might want to remember that. Let me get another one, right? And then he does it. What this tells me, he's able to generate sequences that are essentially novel combinations of experiences. And look, and then he has another one of these sequences, and then he goes there. So make of that what you will, but we actually see several examples of this kind of what seems to be a two-step prediction of him actually pre-playing a sequence which appears to be relevant after the next thing he's done. But I wanted to just focus on this sequence. You saw two nice examples where it starts in the current location, goes straight there and hovers. And that's a novel sequence. That appears time after time, and using the same analysis but just restricted to these trials, these first 19 trials of each day, it's just as good as using the other trials. Basically, the system can generate these good predictions of the future trajectory for novel trials. So that, to me, that's where the imagination bit comes from, because the hippocampus is putting together experience into novel combinations. And I think I was talking to a couple of folks about problem solving today. So this, to me, could be a model of problem solving. So in the interest of time, I'm going to go really quick through this final part, which is just by way of introduction. I won't spend too much time on it, but one of my interests is also to ask the question, well, is this, can you have diseases of sharp wave ripples? Is this a mode of activity? If it's really a unique mode of activity for this kind of exploratory thinking, can it fail? Are we expect it to fail in particular disease models? And I'll just mention it because there is this idea, which is in the imaging literature and in the human patient literature, of something called the default network, which some people may be quite familiar with. This is a number of areas, but includes the hippocampus, that are active when during these sort of rest states. If you like, when you're sitting here, you're kind of zoning out, not listening to me, and your mind is wandering, and suddenly all these areas start lighting up and being active together. And it's interesting because it sort of relates to a little bit to episodic memory. This quote from Mendel Tulving, episodic memory is mental time travel. But it's really been, Randy Buckner at Harvard has described it as a network for free thinking, remembering the past, envisioning or imagining the future, and considering the thoughts and perspectives of others. I had the good fortune to be able to examine an animal mouse model of schizophrenia, which is to say it's a mouse with, it's a calcineurin knockout. It's a mouse that with the knockout of a gene that has some implication in schizophrenia. The story about schizophrenia mouse models is that there is no accepted mouse model of schizophrenia, but there are many possible candidates for genes that can confer risk. And I won't go into the details for reasons of time, but I'll just say we were able to characterize this mouse that really has a plasticity phenotype. It has too much LTP and no LTD in the hippocampus. And what's interesting about these animals is they exhibit 2 to 3 times as many ripple events as normal animals. But what's really stunning is that the place field activity when the animal's running is completely normal. The firing rates, the directionality, various other measures are completely normal. As soon as the animal stops, you get 2 to 3 times as many sharp wave ripples, each of which involves 2 times as many spikes as normal. So the net effect is a more than six-fold increase in excitability as soon as the animal pauses, associated with sharp wave ripples. And then the animal runs and everything's fine. And then we used pairwise measures. I won't go into this, but we couldn't record enough cells in these small mice to talk about replay directly. In this case, we only had 6 tetrodes in the mouse, but we could get pairs of cells, and we could say there should be a relation. If replay is happening, there should be a relationship between how physically the distance between 2 place fields and the spike time separation in these sharp wave ripple events. And that relationship in control animals was all right.
That's Microsoft.
Yeah. But it knows it's just Microsoft. It came back.
Yeah.
That relationship here is completely gone in these mutants. So we think this is interesting, and I won't be able to tell you about it, but we.
So based on this, could you do a similar experiment in a human that's got a hippocampal electrode in place? For example, somebody who's preparing for. No, because I'm not a stupid question.
No, we tried.
Well.
We tried, but we can't get enough cells from them. We will try.
All right, so
At that point, actually.
Yeah, so you're preparing for surgery. You've got a dental electrode in the hippocampus.
Mm-hmm.
Presumably, you can identify a number of individual cells. You do that, right? How many cells would they have to be finding in order to test the hypothesis that there are similar short-wave ripple events in humans?
Well, we're working on it. I'm lucky enough to be able to work with a neurosurgeon at Hopkins, and we're trying something like that. Let me try and scoot. I've gone way over time. I'm really sorry. Thanks to everybody for staying. But the summary. So sharp wave associated place cell sequences can depict recently experienced trajectories and future trajectories. They can predict what the animal's going to do. They can even imagine trajectories that the animal in the strictest sense hasn't done before. So I say here, a bit of a reach. Perhaps sharp wave ripples are a model of deliberative cognition. Okay. They require NMDA receptors during exploration in order for you to see them subsequently. They do not require NMDA receptors just for the generation of sequences if the environment is already familiar. So I think that adds to the idea that this kind of activity could be a model of hippocampal retrieval, and also the idea that that these events, although they can depict novel trajectories, they draw from a structure, a map, if you will, or a set of contingencies between places that has to be learned. And finally, I didn't tell you, but we actually decided to take a comparative approach. In fact, we've been looking at a second model of schizophrenia and finding very similar results. So we're sort of interested in this as a model of a psychiatric disease, and also ways to try and, this is where the human comes in, to decode the content just from local field potentials. So we suggest that sharp wave ripples could be a novel target for studying models and therapies. Well, I was going to show you one more example. Just for the hell of it, yeah. Let's see. So, here we have another event. All right, and the animal goes off back to the food. Now it starts spitting out these sequences. They go sort of willy-nilly. Then he seems to take this kind of behavior. Now he doesn't know where the food's going to be. He stops there, finds it, and he starts spinning. See it? Now that one went sort of through, but in this kind of a shape, and that's it. He finds the food. Okay.
So every shortwave ripple can be just, in this particular animal, every shortwave ripple, well, that's electrically defined, right? Describe some path. You don't see shortwave ripples that are just.
All right. So this is a point of contention with my peers. Basically, we've set a threshold. We said, all right, for some of this analysis, we only want to look at sequences that have a certain sequential structure, and then we don't say where it has to go. We just impose some kind of smoothness to it. For some of the analysis to say, the analysis of, for example, where did these sequences go? Looking for the memory. But if you look at all of them, basically the amount of I don't have the figure here, but if I plot, it really is like this. If I plot the number of cells I've got and the proportion of replays that satisfy this constraint, it just goes up like this across different sessions. So I believe that if we could record that at some point I see no evidence for the proposition that there are any non-sequential sharpwave events.
In a sense, I was sort of mean that he's sitting there, he doesn't suddenly decide to remember something that's not involved in this environment.
Yeah, you can have remote. So you can have remote replay, but they also look like replays. They're also sequential events. But you can have events that relate to other environments. So that has been shown. In my hands, it's actually relatively rare, but you can have it. And then certainly, obviously, in sleep, as we said, sleep isn't really. I'm not monitoring a particular behavioral state. It's really just gone into a different box. It's a boring little enclosure, and then he starts to produce these replays for different experiences. I should just thank several people, members of my lab, particularly Brad Pfeiffer, who's the hero postdoc who did the 250 place cells. He's now he's going to start as a faculty at UT Southwestern shortly. I should thank Xiaoxing and Dahlia for the other data I talked about, my collaborators, and my funding sources. Thank you.
I know it's late, but I'm sure David would happy to take some questions, and then we'll have some reception afterwards on site.
I'm trying to understand, so at first you have these replays which happen in reverse, right? And those, are those really the same thing as the shortwave ripples in this open environment, but the replays in reverse are just constrained by a linear environment? Are they really the same thing? Or?
So they're not, because so unfortunately with the open field, all right, let me back up. So there are several things that place cells can encode. One is position, another is direction. So on the linear track, you actually find that some cells, there might be a cell with a place field here, but it only fires really when the animal's running in this direction. But there might be another cell with a very similar field that only fires or fires a lot more when the animal's running in the other direction. So reverse replay was defined very explicitly as the animal runs in this direction. We look at these cells that fire in this direction, they're firing in reverse. The cells that fire in the other direction, that would be the prospective sequences, they don't fire. And then the bidirectional cells do fire. So that was the basis of saying it was a reverse replay. In the open field, directionality is anyway a bit of a tricky subject. It looks like you probably can, so we've tried to do it, and you probably can encode some information about direction as it happens, but we haven't been able to really say definitively anything about what's going on in terms of, I would expect that you probably do have some events that are more forwards than reverse and vice versa. But one other thing is that you tend to get a lot more reverse replay in a novel environment, which is interesting.
So from what you showed and others have shown, this seems to be a phenomenon that is an active process. That the rat is actually asking himself, well, I'm about to go there, or what if I did that? Or Brady showed like even shortcuts being uncoded, right? So my problem is like from Bujaki or my team, I know that this is initiated from CA3. So does that mean that CA3 is the wheel of the rat, and that what it's going to do after, or is there an involvement of other processes? What is causing these things to happen? Is that just a passive thing? Chishvara was supposing it was just a passive thing because it had just been through the cells. But
Yeah. No, I don't. I think Chishvara is wrong about that. But what I will say is these events are 100 milliseconds long and could be 500 milliseconds long, and seemingly composed potentially of actual ripple events. When you filter for the ripple frequency, you actually see. Sometimes you see these events chained together. And basically, it's. I mean, I should have shown a lot. There's actually, even within these, there may be a sort of pulsing at gamma. So that's a new story. The point is, this is 100 milliseconds, and it's way long. It's much longer than the time it takes for information to get from CA3 to CA1. It's more than enough time to go round through the cortex, back in several times. And Lauren Frank has this new result that. The power of this, so this would be wrong, this is correct. The power of these ripples is modulated at the gamma frequency, so you may have several gamma cycles. Who knows, that could be several spins around the hippocampus, right? So CA3 could play a role in this, but it doesn't have to be all CA3.
So what do you think, who's behind it? Prefrontal with Valerians, or?
Well, for me it's a research program, right? What initiates them, what directs them, what terminates them, what propagates them, and how are they learned, and which synapses, I mean.
And who is there?
And who listens, yeah. I didn't show you, but I mean, there are, for example, I'm sure there are many areas that are modulated by replay, but prefrontal units are strongly modulated. Yeah. Okay. Yeah.
So how do you reconcile this NMDA story? Because why is in the cell? What is the mechanism in the cell that causes them to be affected for recently experienced places versus not being affected for the old ones? You see, it's like I don't understand what.
Yeah.
What
So
is
what
the mechanism at the cell level that is causing this?
So my simple model. So you know that. Perhaps I should have emphasized it, but you know there isn't a topographic map in the hippocampus, right? So there's not a fixed relationship between where cells fire and where they are in the brain. And the reason for that has always been thought to be so that you can get so you can have a whole new map rapidly occurring in a new environment and form essentially arbitrary associations between places. So I do think that's probably what's happening. So I think that when you go in the novel environment, you've got a new collection of cells for each place that aren't particularly associated together, and you've got to put those. You've got to write those associations in the synaptic connections. Who knows? Between CA3 cells, that would be a simple way, but it doesn't have to be. But somewhere, the synaptic changes that encode those contingencies. This place is next to that place. But having done that, you can then run these, you can then retrieve these sequences, even not in precisely the order. You know, as long as you've got the contingencies between places, you can, as demonstrably, produce sequences that haven't been experienced before. So, in a sense, if you want to call it a map, you learn the map through experience, as the connectivity between places. But then your eyes can wander along the map in any way you like.
That's still the NMDA role then.
NMDA to create the map. So you start off with the places. NMDA receptors link the associations between the places, and then a third step is retrieving that information, but in a flexible way which can wander off along different paths in a goal-directed way, but in very flexibly. To me, that's just what the data says. I'm theoretically, I don't know, it's not necessarily what I wanted to find, but I think that's what fits the data best.
Any last burning questions? Slim?
There's wine out there, come on.
Well, we could all have our sequence now head out. to that one goal location out there. Okay, once again, thank you very much, David.